here we go. So we are in our glycolysis, as I mentioned first, and we have glucose entering into the cell. It's going to interact with our hexokinase, and so then that hexokinase is going to make a phosphate group on that, and voila, it has a now phosphate group on that 6, so glucose 6-phosphate. Then that glucose 6-phosphate is going to interact with our phosphoglucose isomerase. Isomerase is going to change the ring. So it won't change how many carbons, still 6 carbons, but it will change the ring. So it's going to interact with that, and then out will come our fructose, a 5-carbon ring, or a 5-membered ring, 6 carbons still. So after that, we have our fructose 6-phosphate traveling into the cell. And as a quick aside, we'll stop there and notice there's going to be a lot of fructose 6-phosphates here. So this will happen continuously, right? And we're going to get a couple of these fructose 6-phosphates. Now, we also have a little bit of insulin because we're working with our glycolysis cycle. So insulin is going to be interacting in the cell, and insulin is going to interact with our phosphoprotein phosphatase, meaning it removes a phosphate group, and it is going to remove a phosphate group from our bifunctional enzyme. So at first, this bifunctional enzyme is going to be active in the phosphatase, but now our fructobisphosphate-2, but then this is going to interact and steal that phosphate, and then that phosphate is going to come off through magic here, of course. Voila, and it comes off, and we have a phosphate group and now an active kinase, PFK-2. So insulin has, moving this back, insulin has interacted with our PP1. It has removed a phosphate from our bifunctional enzyme. And now, whether or not this is the same phosphate, not quite sure, but at least we have a very, a phosphate group in hand in the cell, and the kinase adds a phosphate group to one of our fructoses. So I know I'm getting a little cheated out here, but one of our fructoses. So that interacts and makes fructose 2,6-bisphosphate. Now, we have fructose 2,6-bisphosphate, which was made by our kinase here, PFK-2, and this is because it's on the 2nd carbon of the fructose. And fructose 2,6-bisphosphate is going to allosterically interact with our PFK1. Notice the number difference. It is a tetramer, so that's why I have it drawn here like that. And so it is going to interact, make this more likely to do its job, which is take one of our very, many fructose 6-phosphates in the cell and change it into a fructose 1,6-bisphosphate. That fructose 1,6-bisphosphate is changed by our PFK1, and so allosterically activated by our fructose 2,6-bisphosphate, and this is to show that there are a lot of fructoses happening right now. This 1,6 goes on to interact with aldolase, and it becomes then a 3-carbon DAP and GAP, and those move on into the cycle. this continuously goes on. This 2,6-bisphosphate still is interacting. Remember, it's also allosterically modulating this phosphatase. And so this is no longer active because this 2,6, which there are plenty of, is going to shut that down. All right. So that's why it's a bifunctional enzyme. And then, of course, it continues. It uses its ability. Fructose kinase is going to make a 6-phosphate fructose become a 1,6. It's going to continue on, interact with aldolase, and become a 3-carbon DAP and a 3-carbon GAP and a 3-carbon DAP. Those will go on to the cycle, and it continues. So I hope all in all, now you see that there are many times where fructose 6-phosphate is transformed. And because we have plenty of it, that's why we can interact with this enzyme and continue down our glycolysis pathway. All right. Take care, everyone. Bye.